Hari Om, children. My name is Iswarya. Today we are going to see a glimpse of heredity and evolution from class 10. So, shall we move on to the session? Yeah. Okay. So first, I just want to clarify a few things. What is the difference between DNA and chromosome? Come on, I want answers from everyone. Let it be an interactive session because since you are a little more known to this topic, it will be a little interactive. What is the difference between chromosome and DNA? Okay, see, we usually use these terms. What do we use these terms? Chromosome and DNA. What part of biology usually comes? Yeah?
DNA is deoxyribonucleic acid.
Okay. And what about chromosome?
It is a thread-like structure.
Okay. Where is it found?
Nucleus.
Which nucleus? Everybody can try.
Nucleus.
Just try. Yes, which nucleus? Which nucleus it is? How many nucleus do you have? If I ask you, how many nucleus do you have in your body, what will you tell?
It's one in each cell.
Very good. It is one in each cell. Yeah, except your RBCs. All other cells in our body have nucleus, and it is very, what is it called? Very excited to think that every nucleus inside every cell of your body has the same set of chromosomes. And hope you know how many set of chromosomes do we have? How many chromosomes do we have in our body?
23 pairs.
23
Very
pairs.
good. 23 pairs. That is 46 chromosomes which are found in 23 pairs. Correct. So if you take a cell from your skin, what is the number of chromosomes in that nucleus? Skin cell? Obvious. The answer which you gave now. Yes.
46.
46 chromosomes. Okay. If you take a cell from your heart?
Same.
46.
Same. Okay. If you're going to take a cell from your stomach? Same. Correct. If I'm going to take a cell from your ovary? Yes. If it is from the ovary? Same. Is it? Just think. Okay. We'll imagine like this. What is the female gamete?
Ovum.
Ovum. Okay. Who is producing this ovum?
Ovary.
Ovaries are producing ovum. Correct. Okay. What is the male gamete?
Sperm.
Sperm. Who is producing it?
Testes.
Whereas how sperm produced? It's produced in the testis.
Testis.
Correct.
Testis.
Okay. So you all know that the zygote is formed by the combination of a sperm and an ovum. Am I correct? Yes. So we'll imagine like this. The skin has 46 chromosomes. Your stomach cells have 46 chromosomes. Your kidney cells have 46 chromosomes. And if you say your ovary cells or the germinal cells which are present in the ovary also have 46 chromosomes, you are going to combine the ovum and the sperm. So 46 plus 46, what is the number of chromosome you get? What number of chromosome you get? 92. Am I correct?
Yes.
Yes. So imagine in a human cell, you have 46 chromosomes. And a zygote which is coming out of a human cell, if it is going to have 92 chromosomes, will you expect that to become a human child or a normal child? No. Am I correct? So there should be something which will happen so that you can have the correct number. What can we do for that? Yes, Chinmayi is something about to answer. Yes, Chinmayi. Come on, let it be. Just tell. What should you do?
I'm not sure, but
That's
maybe
okay.
it will be only half chromosomes.
Very good. That's it. That's it. So if you have half plus half, it becomes 1. So instead of 46 chromosomes, what do? We get 23 chromosomes from the mother, 23 chromosomes from the father, and that's how we get 46 chromosomes. And the rest story, you know, the zygote continuously gets divided, multiplication, and then they turn into embryo. Embryo turns into fetus. Fetus turns into the infant. Am I correct? The rest of the story, you know, we have studied in how do organisms reproduce. So, in short, what you get from your mother and father, how are you getting it? Through our who is bringing your mother and father's characters to you? Now you should tell me. Who's bringing it? The sperm represents your father and the ovum represents your mother. Am I correct? So this ovum and sperm are cells. First, that we have to understand. So the cell called sperm and the cell called ovum will combine to form a cell, single cell called zygote. So what is the specialty of here is this will have half the number of chromosomes. This again will have half the number of chromosomes. They together form the complete set of human chromosomes. That's how the zygote gets 46 chromosomes. So in short, it is obvious to understand that something which is transferred from the parent is what gives us the character of our parents. So what is transferred here? When we studied fertilization, you have studied what is the fertilization process? What is actually combining? What is actually combining there? Pardon?
Male and female gamete.
Okay, male and female gamete. So if you see it little more detail, you will understand that not the sperm as such fuses with the ovum. The nucleus of the sperm will enter inside the ovum. Okay, so the nucleus of the ovum and the nucleus of the sperm will combine. That is how we get 46 chromosomes inside the nucleus of the zygote. Okay, now tell me who is bringing the character of your father and mother?
Genes.
Where is it present? What came from your father and what came from your mother? Just now you told.
Chromosomes.
Chromosomes. Where? From the nucleus. Inside the nucleus of the sperm and inside the nucleus of the ovum, you have the chromosomes. So these two chromosomes will combine and form the chromosome set of that zygote. Okay, then you know about binary fission. Just the photocopies. That is what happens in the initial stages of the division or multiplication of zygote cell. And then embryo is formed, then later it develops into different parts of the body. So in short, whatever you are getting from your father and mother is got through the germinal cells. Okay, that's called germinal cells. 1 minute, let me present. Okay, is the whiteboard visible?
Yes, ma'am.
Okay. Yeah. Okay. So germinal, Germinal, germinal. Okay. So the germinal cells, is it visible, children? Yeah.
Yes, ma'am.
So germinal
Yes.
cells are the special cells which will not have the whole set of chromosomes. What do they have? They have the half number of chromosomes. That is, they'll have 23 chromosomes in them. Okay, there is another set of cells in our body. What are they called? Do you have any idea? The cells which are there in your skin, the cells which are there in your heart, the cells which are there in your kidney, they are called somatic cells. They're called somatic cells. Okay, so somatic cells are the cells which are found in your body. So except your ovary and testes, the whole body is made up of cells called somatic cells. So how much number of chromosomes are present in somatic cells? You can tell it now. Yes? 46. Correct? 23 plus 23, 46. So if you check the nucleus of somatic cells, it will have 46 chromosomes. And if you check the nucleus of germinal cells, it will have 23 chromosomes. Okay, now that this is clear. Now, another question for me. So you can see if it is a nucleus. Okay, let me consider this as a nucleus. And you have something like this. And you have a nucleolus. Am I correct? This is what we have studied. What is this part called? It has a name. What is it called? Inside the nucleus, the thread-like structure, which is called, what is it called?
Chromosomes.
No.
Chromatin.
Pardon? Pardon. Correct. What is it called? Chromatin. Am I correct? It's called chromatin. Okay. So what is the difference between the chromatin and chromosome here? See, if you take a normal cell, you will have the chromosomes in the form of chromatin only, like a woolen ball. Okay, but at the time of cell division, when the cell is ready to divide, these structures which are clumped up, they will form a structure, a unique structure that you know. What is this called?
Chromosome.
What is this called? Yeah, exactly. This is called a chromosome. So, chromosome and chromatin reticulum are the same. What is the difference? Chromatin reticulum is a structure which is found when this nucleus or the cell in the normal stage, normal functioning stage. When the cell is about to divide, they take up the chromosome structure. Is that clear now? Okay, so this is about chromosomes. Now tell me what is, now you understood what is chromosome. In this chromosome, where is this DNA present? And we have another term called genes. DNA is there, genes is there. So confusing. What is chromosome? When we ask genetic material, we say chromosome. We ask genetic material, we also say DNA. When we ask genetic material, we also say genes. Am I correct? So how are they same? Now you understood what is chromosome and chromatin, right? Now tell me what is the connection between this DNA, gene and the chromosome? Yes? Okay, I'll show you a picture so you can infer from it. But just give me 1 minute. Let me share the other screen. Okay, is the screen visible, children?
Yes, ma'am. Yes, ma'am.
You? Okay. Yeah, so this is the structure. Can you see the chromosome here? Yeah?
Yeah.
This chromosome is having something. I'll show you one more picture that will be more evident for you. Yeah, this one. So this is the exact one. So this is the chromosome inside the nucleus. If you unwind, that's called unwinding. Just like pulling a thread from the woolen ball, you're just unwinding it from the chromosome. You find structures like this. So it's like a button, right? There's a button and there's a thread wound on it. Am I correct? Yes. So these buttons, these ones are called proteins. They are proteins, special proteins they are. Okay, that you'll be studying in the later classes. So this proteins along with this one is your DNA. So this DNA will be wound in a special protein. Is it visible now?
Yes.
Yes. So this DNA, which is bound in a special protein, is forming this particular part. Is that clear? Is it okay now? Do you get it? Yeah?
Yes.
Should I explain once again?
Yes.
Is it clear?
Yes, ma'am.
Okay, fine. So this DNA is the one which is wound with these proteins, which is forming the structure, this chromosome structure. Okay, now DNA and chromosomes clear, ma'am. Now what is gene? Now what is gene?
It is a part of chromosome.
Okay. So even DNA, if you take a DNA, that's also a part of a chromosome. Then what are you going to tell about DNA and gene now?
It controls characteristics
Okay, almost there.
in organism.
Almost there. Anything else?
It is the functional unit of heredity.
Okay. See, this is a DNA. It's a double helical structure. We know. Am I correct? So this is a long DNA. You know, there is a project called Human Genome Project. There was a project called Human Genome Project, which sequenced the whole set of DNA. It took more than 10 years to sequence the DNA of human. Okay, so this DNA is a single thread. Now, in that, if you are going to take a small portion like this, and this portion, if it is going to code for a character, that's called a gene. What is gene? So it's a fragment of DNA. Fragment of DNA coding for a particular character. Coding for a character. Coding for a character. Okay, it can be any character. It can be your skin color. It can be your lens color. You can be a height. Whatever character is given from your parents or you take it from your ancestors, that character will be in a gene. Okay, one particular fragment of DNA will be responsible for that character. And that fragment is called a gene. Now it is clear? Is it clear now? So you understood what is chromosome,
Yes,
what is DNA, what is gene. Okay. See, why I said this is these are basics of the very beautiful topic what we are studying. It's genetics. Okay, genetics is a branch of science which deals with heredity and evolution. Okay, so what is heredity? Heredity is a very familiar term for all of us, right? Even a layman, a normal person who knows nothing about biology, they use this term heredity. If you have some character which is resembling from your mother or father, immediately we get that comment. You are just like your father, you're just like your mother, you have got it from your father, got it from your mother, correct? They don't even know what is DNA, what is gene, but they correctly say that this is from your father, this is from your mother, or this is from your grandfather. Or sometimes they say your whole hierarchy is like that. Am I correct? They say, is it not? So this is a very interesting, actually a very interesting truth of the nature, that not only us, every living organism, we know every living organism have a chromatin material. Even we have a difference between prokaryotic and eukaryotic organisms, correct? Am I correct? They are not the same. Bacteria and the eukaryotic organism is not the same. But what is common among them is the genetic material. Even a virus, which is always a controversy of whether it is living or non-living. It is always a controversy. Yes, even that virus has a genetic material. It can be an RNA or a DNA. Is it not? We know. So that especially at this crucial period of corona, we know a lot about virus, correct? So now we came to know that virus is also having DNA as we have. Virus is also having a genetic material like we have. So you have to imagine, children, even a small organism which is not even the size of bacteria, that also has a genetic material. So you have to understand how important this genetic material is and what is it doing to us. Okay, now, okay, the gene has come to us from the father or mother. You have got a gene. How is it expressing out? How is that? For example, your mother's hair is a long hair. Okay, you also have got that long hair from your mother. How did it get? How did it come? You say that it came from gene. Okay, what will that gene do? What will that gene do? There is a process called protein synthesis. Every gene will be coding for a protein. Okay, that's what I said, a fragment of DNA which will code for a protein. So that protein will induce this particular character. Is that clear? Yeah. So, but there are certain characters which cannot be passed through this gene. Now, in the genetic term, we call it traits. Traits, traits. Okay, it's called traits. 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 Traits means characters. Okay, so there are so many characters we have from our father and mother, and there are so many characters which your father and mother cannot give it to you. For example, your father knows swimming. You cannot inherit from your father. Okay, your mother knows driving a car or driving a motorbike. You cannot inherit from your mother. That you have to acquire during your lifetime. So based on this difference, there are two types of characters or two types of traits. First one is inherited trait. Inherited traits and acquired traits. Inherited traits and acquired traits. What is this inheritance? Inheritance is nothing but heredity. So inherited traits are the ones which you get from your father or mother. Inherited means which you get from your parents or inherit from your parents. Now, what is acquired trait? Just now I said. What is an acquired trait?
Character acquired in a period of time.
Very good. Character which you have to acquire in your lifetime. It will not get not inherited. It is not inherited from your parents. So these traits, these acquired traits will not pass from one generation to another, whereas inherited traits will pass from one generation to another. So how many types of traits are there? Can you show me your hands? How many types of traits are there? Two types of traits. What are they? One each. Anish, what is it?
Inherited and
Very
acquired.
good. Inherited. What is inherited trait?
From parents. Traits from parents.
Okay, traits or characters which you get from your parents through your genes. Okay, now the next one. Anyone else? What is the other one?
Acquired traits.
Acquired traits. Acquired traits are the ones which you will not acquire from your parent. You will not acquire through the gene. Then how will you acquire it? Through the lifetime. Okay, example. Just now I told you. What is the example of acquired traits?
Swimming, driving.
Okay, acquired traits example, swimming and driving. Okay, now inherited traits, can you give me examples?
Height.
Very good, height. Okay, then
Eye color.
eye color, very good.
Skin color.
Skin tone, skin tone, skin tone. Okay, so these are all traits which you get from your father or mother. Okay, so these are the two types of characters. Either it will be acquired or it will be inherited. Is this concept clear? Okay, now we understood two basic concepts of genetics. First one is the what is chromosome, what is DNA, and what is gene. Then we started to know characters are inherited from the parent or you will acquire in your lifetime. These two concepts are clear? Okay. Now, any doubts in this? Can we pass on to the next one?
Yes.
Yeah.
Yes.
Okay. Now, genetics is a branch of biology which deals with heredity and evolution. Okay. So this genetics is a very vast field of biology, and there are so many people who have contributed for this particular subject. Okay, out of which there was one person called Mendel. His full name is George Mendel. He was an Austrian. You know what's Austria? Yeah, you know the country Austria. So he was an Austrian monk. See, those days, education were always connected to the religious centers. So he was a monk in the church and he was looking after the convent garden. Okay, it's a very good story. You have to listen and you have to get inspired. There are so many people in science who can inspire us a lot. So this person, he was working in the garden and he was very much curious and he was very much longing to have higher education. Okay, but since certain restrictions were there, he was in a position to take care of that particular convent, that particular garden, and he was there doing its small research works and doing small studies and all those things. So the garden was under his control. He used to observe plants a lot. He loves botany, so he used to observe plants a lot, and he used to find characters in them. He used to think about it, and the very common plant in his garden was pea plant. Okay, so pea plant has two varieties. Can you tell me the color? Two types of pea you get?
Green.
Green.
Yellow.
Yellow, very good. So green and yellow. There are two varieties of pea plant in his garden also. And he found there are so many characters which are contrast. Okay, for example, is what I said, the seed color. He saw some plants with green color, some plants with yellow seeds. And he saw that some plants, the seeds were proper round. And in some plants, the seeds were shrinked, wrinkled. Okay, he saw that some plants were tall and some plants were short. And he saw that some plants were giving purple flowers and some plants were giving white flowers. So lots of difference in one particular plant itself. So he was very curious to know what will happen if I combine these two. Okay, so usually I'm asking, if you combine black and white, what is the color you get?
Black.
Can you combine a watercolor? We say watercolor.
Gray.
Yeah, watercolor. Yes, correct. Gray. Yes, we get gray. Am I correct? If I going to combine blue and yellow, what is the color you get?
Green.
Green. Very good. So you all have a very good artistic taste. Very spontaneous answers I get. Okay, see, green is a combination of blue and yellow. So you get a proper blending. Am I correct? And again, if you are going to take the difference in proportion, accordingly the color changes. Is it not? So anything of two different or two contrast, if you combine them, what is the expected result? A moderate one, a middle one. Yes. So Mendel was also expecting the same. He thought, okay, let me combine. He first took the height of the plant. I said there are two characters. One plant is tall and one plant is short. So what he thought, okay, I'm going to combine these two. And you know how it is combined, children? Artificially, how it is combined? Any idea how it is done? It's a very old practice. He was a person in 17th century. So he have did it. What is that done? Okay, how is naturally sexual reproduction occurring in plants? Naturally, how does it occur? Yes?
Pollination.
Very good, pollination. The pollen grains carrying the male gamete will fly, or it will be washed through water, whatever it is, the agents of pollination, and it will go and fall in the stigma, which is a part of female. Am I correct? From the stigma, it comes down, it fuses with the ovum, which is present, or the egg cell, which is present inside the ovary. And the seed is developed, fruit is developed. Again, the seed falls and it develops into a new plant. That's how it usually happens, naturally happens. Yes. So now what can we do it if it has to be done artificially?
Vegetative propagation.
No. In vegetative propagation, it is single parent, right? How will you combine? Yes?
Grafting.
Okay. Again, grafting, it is among the somatic cells. Correct. So you don't have a probability. So it is called artificial pollination. We will be studying in higher classes. What you do is he's not a very expert botanist. So what he did is he just took the pollen from the male plant and which was considered as male plant, and he just dusted it in the small female plant. Okay. So, and it was wrapped. This is a usual process. Then after that, the resulting, what do you expect? What will it be? What is supposed to be? What is it supposed to be? If you're going to combine tall and short, what is it supposed to be?
Tall.
That's okay. That since we studied Mendelian experiments, we know it is tall.
Medium.
Actually, it is supposed to be medium, correct? An obvious answer, a logical answer is if you are going to combine tall and short, it will be medium. So even Mendel was expecting a medium plant. But to surprise, what was the plant which he got? Now
Tall.
tell me the answer.
Tall.
Tall. Okay, so he was wondering what happened to that short. Okay, so he was very curious. So now next step, he had a one more try. So what he did, he combined this generation, that's the offsprings, the young plants which he got, baby plant which he got. He took again 2 plants from the next generation and he combined among them itself. Okay, and he got the next generation. Now to his surprise, he saw the small plant. He was wondering, this particular character of small dwarfness was not seen in the previous generation. It was there in the parent plant, but in the middle generation, it was not found. Again, it has appeared in the third generation, that is the second F2 generation. Okay, so he was wondering, how did it happen? Then he understood that something is there which is controlling this particular process of taking what is it called, getting characters. He understood that there is something which is controlling that, and he named it factors. It's called factors. He didn't name it gene. He was not the person who named it gene. It is factors. Okay, so factors are it was the term which was given by Mendel, and he said that there is something called factors which is present in every living organism, which is transferred from one generation to another. Even though he didn't quote it as gene, the rest explanations were as such about gene. So he said factors are something which is transferred from one generation to another. Then he started to make a flow chart. So what did he do? He understood that this tall plant is dominant because it is suppressing the dwarfness, isn't it? When you see, you combine tall and dwarf and you are getting tall. So who wins the race? Tall. Tall wins the race, correct? So he considered it as a dominant one. Okay, then what he did is he represented it as capital T. Okay, before this, you know, a gene is always coming in a form called allele. Have you heard this term allele? Yeah. What is allele is? See, a gene, for example, we'll consider tall. Okay, tall is a character of a plant. There should be a gene which is coding for this character. Always we will represent a gene as a pair allele. Why, can you tell me? Why is that? This is what it will be present in the parent. Okay. Always this answer for this lies in the introduction which I gave you. I gave the introduction for this purpose only. This one is having a pair of allele, and a pair of allele gives to a gene. If you see a gene, if you take a gene, any gene for that matter, every gene will be represented by a pair of allele. Every gene will be represented by a pair of allele. For example, I said capital T, capital T. You have studied sex determination in 8th standard. Am I correct? How is the gene of female represented?
XX.
XX. Am I correct? How is the gene of male represented?
XY.
XY, exactly. So by this time you should understand that any character, this is the character of sex. Am I correct? This determines the sex of that fetus or the baby. So any character for that matter will have a pair allele to represent it. Did you understand? This one is called an allele. Okay, so he understood that there is a double character. So what he did is he made it as capital T, capital T. Since this dwarf didn't express, it didn't win the game, right? So it was made small t, small t. Okay, so capital T, according to Mendelian experiment, what does it denote? What
Tall.
character? Tall.
Tall.
Okay, very good. Now, small t represents
Short.
short or dwarf. Okay, now we are going to combine these two. How will we combine? Will we take capital T, small t, or something else is there? This I want to,
One character.
Why?
take
Why?
one type,
Why
one
you
one.
should take only one T? Why? Correct. Answer is correct. You will take only one T. Correct. Why?
Both characters should be combined.
No. Anything else? This is the parent and this is the gamete. So inside the gamete, it will be, what should we tell this? It will be half the number of chromosomes. Am I correct? Here, if it is 2, it's denoted as 2n. In the parent, if it is 2n, here it will be
N.
Very good. N. Okay. So only then it will combine and form something like this. Am I correct? So capital T, small t forms. What is the product? Spell it out. What is it?
T.
Capital T, small
And
t.
small.
Am I correct? So this is called a hybrid. Do you agree? Do you agree it is a hybrid? Why is it called a hybrid? Why is it called a hybrid? Hybrid is a very common term. Your grapes, seedless grapes, which is purple in color. Usually purple grapes, a normal grape will have a seed inside it. Am I correct? So the other one which you get in the market, seedless, that's a hybrid variety. What is hybrid there? They combined the plant of green grapes and the purple grapes. So the resulting will have the purple taste, but it will not have seed like the green grapes. So that's a hybrid. Okay, it's a very common term. So hybrid means a resulting product from two pure breeds. When two pure breeds are combined, it forms a hybrid. Is this clear? Okay. Now he understood. Now what did he see? The resulting generation. This is called F1 generation. So F1 generation, when he looked, what was it? Was it tall or short? Tall. Okay, so it was tall. And what did he do? He was wondering where did the dwarf character go? Is it not? So for that, what did he do? I told you, what did he do? He combined. What did he do?
Generation.
He combined the F1 generation. So he combined capital T with capital T, capital. Am I correct now? Two parents, both are hybrids. But again, how should it come down? How should it come down? Probability, you tell me. This capital T can combine with this capital T. This capital T can combine with this small t. This small t can combine with this capital T. This small t when combined with this small t. So what is the answer here?
Capital T.
Capital T.
Capital T, capital T. This one?
Capital T.
Capital T.
Very
Small
good. Capital T, small t. This one?
Small t.
Small T. Capital T.
Small t, capital T, but still, just like your English, capital T will come first. Okay. Then here?
Small t. Small
Small t.
Very
Small.
good. Small t, small t. Now tell me, how will they appear? Genetic genes, let the genes be there. How will you represent this? By seeing, what will it be? This one.
Tall.
Tall.
Very good. Tall. These two?
Tall.
Tall. Tall. This one?
Short.
Short.
Why?
Dwarf.
Why?
There are two small t's.
Both are.
Oh, so the small t will represent when it is getting another small t. Is it? So you came to that conclusion. Correct? You understood when you saw this, you understood that this particular capital T is very dominant. Even it is not having another capital T, it will represent. It will suppress this small t and it will represent. Whereas the small t is a poor child. It needs another small t to have a small company to get that confidence. Okay, so this small t will express only if it gets another small t. Is that clear? Now, if you see this, what is the ratio? What is the ratio? How many tall are there?
3.
3. How many dwarf?
1.
1. So if you see, when you see the plants, F2, this is F2 generation. If you see the F2 generation plants, physical appearance, 3 were, this is just a ratio, okay, not that only 4 plants came out. Out of 4 plants, 3 were tall and 1 was dwarf. Okay, so this is by seeing. But if you take the genes, they are not same. Correct. For example, if you take the first one, it is capital T, capital T. This is one type. Am I correct? So this is one type. What are these two? Capital T, small t. How many capital T, small t are there?
2.
2. So capital T, small t is 2. How many small t are there?
1.
1. So there are 2 ratios you have to learn in this. First ratio is 3:1 and the second ratio is 1:2, 2:1. Now this ratio you concluded by seeing the physical appearance of the plant. So this ratio is called as phenotypic ratio. Phenotypic ratio. And this ratio you made using, what did you consider when you make this ratio?
Genes.
Genes.
Very good. Gene, the genetic makeup, correct? So they are called as genotypic ratio. Is that clear? Yes. So there are 2 ratios you will conclude with the experiment. 3:1 by seeing the plants and 1:2:1 by observing the genetic makeup of that particular plant. Is that clear? So likewise, Mendel tried with flower color. He tried with seed. When he tried with seed, yellow was dominant. So here you have to conclude that what is the dominant allele here or dominant character is the one which is represented in which generation? In which generation? 2 generations we saw, parent, F1, F2. In which generation dominant will represent?
F2.
F1. In F1 itself, you can understand which is dominant. Okay. So the character which represents itself or express itself in F1 generation is considered as a dominant character. Once again, what is a dominant character? A character which is expressed in F1 generation. That is first point about dominant character. What is the next point? What is the speciality of this dominant character? What is speciality? It's very strong. So dominant allele does not require its pair allele to express. Right, does not require its pair allele. Example is T for tallness. Okay, so tallness is a dominant character. So because this capital T did not require another capital T to express itself. Even it had small t, it doesn't matter for it. It will represent. Whereas there is another character which needs its pair allele to represent itself. What is that character called? It is called recessive. Okay, so this character needs its pair allele. Needs its pair allele to represent. Is that clear? Okay, so we have studied 2 types of characters with respect to 2 genres. First one, with respect to whether they are inherited or not. They are called inherited traits and
Acquired traits.
Acquired traits. Very good. And whether they are strong or whether they are expressing in the F1 generation, with that they are classified into 2. What are they?
Dominant and recessive.
Dominant traits and recessive traits. So dominant traits are the one which will express themselves in F1 generation, and they are strong enough that they don't need their pair allele to represent. Whereas recessive allele is very weak, they need their pair allele to represent themselves. Example here, what is the example I can suggest? Small t, which represents the dwarfness. Okay. Okay. So this is one example of experiment what Mendel did. So Mendel did a lot of experiments and he had contributed a lot to genetics. That's why he is called the father of genetics. So who is the father of genetics?
Mendel.
Mendel. John Gregor Mendel is the person who is the father of genetics because he has done a lot of contribution to lay a very good foundation of genetics. Above that, we have a lot of improvements going on. We are technologically so sound. We are even trying for so many things which was not even a thing to imagine. That's only because of the contribution of such great scientists. They have contributed a lot so that we can understand the basics. Children, you have to imagine, now we have so many equipment, so many the social media exposures, we have so many books to study, we have people who have known it, we have resource people to explain to us. But those days when these scientists were curious and they were behind so many concepts like this, they didn't have any aid. It is all from their what is it called, conscience, or from the insight they got all those things, which is all was subtle. From the subtle mind they got all those things, and they have contributed a lot to the different fields. So Mendel is a person who have contributed a lot to genetics, and that is why he was called the father of genetics. He is the father of genetics. Okay, now based on all these experiments, he proposed 3 laws. Okay, I'll just name them. First one, can you name? Do you know?
Law of dominance.
Law of dominance. Okay. Then?
Law of segregation.
Law of segregation.
Okay, law of segregation. Then?
Law of
Law of
independent
independent
assortment.
Very
assortment.
good. Law of independent assortment. Now, this law of segregation and law of dominance was proposed based on the experiment what we saw now. And that experiment is called as monohybrid cross. Monohybrid cross. Cross means, you know, 2 different things are combined. Cross breeds are very common in your dog, very common in your pet animals. Correct. So a cross is something which happens between 2 different things. Now, monohybrid cross. Can you explain it now? Now you can explain. If I would have taken the name first, you may not know what it is. Now, why is it called monohybrid cross? Now tell me. You know the experiment.
Only one character is considered.
Very good. Since we consider only one character, it is called as monohybrid cross. Okay. So this monohybrid cross, based on this monohybrid cross only, he proposed law of dominance and law of segregation. Okay. Now, there was one more doubt for him. He was wondering, before passing on to that, your sex determination is a part of monohybrid cross. It's also an example of monohybrid cross because we consider only one character. What is the character we consider? What is the character we consider? Sex of the baby. Correct. We are going to consider the sex of the baby, or the whether it is a male or a female. How we are going to do that? We are going to take the gene from the father, XY, and we are going to take the sex chromosome from the mother. What is it? X. Okay. Now this is my father and mother. In the gametes, what will happen? What are the gametes? Now you took capital T, you took one capital T. Small t, you took one small t. Likewise, what will you take here? What will you take here? Here, what will you take? That you tell me.
X.
Good. X. Correct. Then here, what will you take?
XY.
X and 1. Okay. So, if a father's sperm is having X, this X will combine with this X and form XX, which
XX.
means the baby is a
Girl.
Female.
Girl.
male. Very good. Now the father's gene, the sperm is going to carry Y. So this Y will combine with this X, and what will it form?
Boy.
XY. So it
XY.
means it is a male. Okay. Now tell me, what is the probability of a male or a female? What is the probability? I'm asking whether it is a boy or a girl. What is the probability? What will you tell? Very simple. What is the probability?
50.
50-50. Correct. 50-50. So it's a 50% probability of the fetus turning into a boy or a girl. That is 1:1 ratio. There we saw 3:1. Correct. Here it is 1:1. Now, who's determining the sex then?
Father.
The chromosome present in the sperm. This chromosome present in the sperm determines the sex of the fetus or the baby. If it is X, it is female. If it is Y, it is male. Okay. See, when this is a very usually asked, very common question in your board exams. And this is asked either in 2 mark or 3 marks. Sometimes it comes as a subdivision in 5 mark question also. So it's a very important question. So this particular question, when it is even is asked in 2 marks or 3 marks or a subdivision of 5 mark, these 3 points are very important. This has to be there. 50% probability or 1:1 ratio. Chromosome, if it is having X, it will turn into female. If it is having Y, it is turned into a male. And last point, sex of the baby is determined by the father. That's very important. Sex of the baby is determined by the father. Okay, so this is also one kind of monohybrid cross. Now, this monohybrid cross, we'll just put it into a table. I'm just drawing a table and I'm going to make it. What is the symbol, children? What is that symbol? This one. Have you seen the symbol anywhere? These 2 symbols?
Yes.
Have you seen? No. Yes or no? No.
Yes.
What is it?
Represent male and female.
Very good. Which represents male? This one represents
Male.
male. Very good. This represents male and this represents female. Okay. So I'm going to represent this as male and female. I'm going to divide it into 4 boxes. Capital T, small t, capital T, small t. Now what will come in this box?
Capital
Capital T, capital.
Okay, what will come in this box?
Capital T, small.
Capital T small.
What will come in this box?
Capital T, small.
Capital T, small
Capital
t.
T, small t. What will come in this box?
Small t, small t.
Small
Small
t.
t, small t. So have we cross-checked 3:1? Have we cross-checked 1:2:2? Sorry, 1:2:1? Yes. This one is called Punnett square. Punnett square. Okay. Punnett square was an easy method which helps us to to determine the genotype and phenotype, and that was discovered by a person called Punnett, and it's named after him. Now, in monohybrid cross, it doesn't have much significance because monohybrid cross it's very easy because one character only we are going to take, and maximum probability is only 2. Either it will be a tall or a dwarf plant. Yes, and if you see the genetic makeup, it is we can easily understand. But this will be very useful for the next experiment what Mendel did. I'll just explain you the experiment. when he was observing, he found that, till now is it clear, children? Monohybrid cross is clear? Yes. Okay. So when he saw the seed color, if it is yellow, it will always be round. And if the seed color is green, the seed will always be wrinkled. And he understood by this monohybrid cross that yellow and round are dominant. Am I correct? By the end of monohybrid cross, he understood that yellow is dominant and round is dominant. Sorry. So he want to understand whether this yellow and round will express if they are separated. That is, he was thinking maybe this yellow will represent only if it is combined with round character. If I am going to separate this yellow from the round character, it may not express. Maybe they are dependent. So he want to clarify this particular doubt. For that, what he did is, instead of considering one character, he considered two characters. What are the two characters he considered? What are the two characters he considered? Seed.
And color.
Very good. Seed color and
Shape.
very good. Seed shape. So instead of one character, now he considered two characters. So how should I name this cross now?
Dihybrid.
Dihybrid. Since he took two characters, it's called a dihybrid cross. Okay, so he was ready to take these two characters. Now we'll consider them. How will you write? How will you represent round? Round is dominant. So what is the initial you have to give? Capital R. Very good. That's the privilege they get. Capital R, and yellow is dominant. So what is the privilege you will get? Capital Y, capital Y. So this is yellow round. Now green wrinkled, how will you represent?
Small r and
Smaller.
Oh, very good. Yeah, you have to write it small r, and yellow will be small y. Sorry, green will be small y. Okay. Now this is parent. You are going to combine these two. What will you get here? Gametes. In gametes, what will it be? In gametes, what should it be?
Capital R, capital Y.
Very good. Capital R, capital Y. Here,
Small r, small y.
small y. Very good. Now, what will be the combination? Obvious, we know. What is the combination? Capital R, capital Y, small y. And we already know what is the expected hybrid. We know what will be. If you see that F1 hybrids, you know what it will be. What it will be? Round and yellow. This was already known to Mendel. It was obvious because by this time monohybrid cross got over, he understood that dominant character will represent in F1 generation. Now, what is his doubt? If I am going to separate, when will it get separated? Only in the F2 generation. Okay, so he wants to separate these two and know whether they will express or not. Now, so for that, what should we do? What should we do? Whom should we cross? Whom should we cross? F1 hybrids. Correct. So I'm going to cross the F1 hybrids. Capital R, capital Y, small y will combine with capital R, capital Y, small y. Now, this can't be drawn like this. What we did for this monohybrid cross, we just combined. That cannot be done. This is where you have this Punnett square. Okay. So when he, okay, first we'll finish what happened. What, can you tell me what would have happened? What would have happened? Do you think it would have got represented, got separated? Yes? Yes, it got separated. So it was a usual combination of RY, then another new combination of capital R, small y, and small r, capital Y, and the other pure variety RY. Now this is round yellow. Now tell me, what is this one? What is this one?
Round and green.
Round and green.
Round green. Very good. What is this one?
Wrinkled and
Wrinkled yellow. And what is this one?
Wrinkled green.
Wrinkled green.
Wrinkled green. Now, there are two combinations. They are called two combinations. What are the two combinations? These two are called, what are these two? They are, what are they? Pure. Correct. Yes, they are called parental combinations because this was found in the parent. Parental combination. And what is these two?
Hybrid.
New
Hybrids.
combination. New combination. Hybrid already, F1 itself is hybrid. So parental combination, new combination. So he controls, so he understood, okay, I understood that this round or yellow does not require itself, the both, to combine or to represent. That is, yellow is dominant even if it is not present with round. Round is dominant even if it is not present with yellow. Okay, and based on this, he proposed a law called law of, what was the law?
Independent assortment.
Independent assortment. What is sorting means what? Assorting means what? What does assorting means? You know what is assorting? Assorting. Assorting means getting separated. Okay. Now, independent assortment means, according to this law, Mendel says that the dominant characters can assort and represent themselves independently. The two dominant characters which are together can assort and represent themselves independently. They don't require another dominant pair with it. Do you understand? So this was the law he proposed based on dihybrid cross. Now we have to draw the Punnett square for this dihybrid cross. Can you just draw it for yourself also? So I'm just making a table with two different characters. What are the two different characters? You tell me. What are two different characters?
Color.
Yes. So I'm going to have
And shape.
1, 2, 4. So the same one, male, female. There I represented capital T, small t. Here, how will I represent? 4 combinations you have to represent. Capital R,
Capital Y.
Capital Y.
capital Y. Very good. Here, capital
Capital
R,
R, small y.
small y,
Small
small
r,
r,
capital Y.
capital Y,
Capital.
small r,
R,
small
small
y.
y.
Again, the same one here. Capital R, small y, capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Now, fast, we'll just tell the character alone. What is this? Round yellow. I'm
Round.
just giving the, this is also round yellow. Correct? This one is also round yellow. Just say R, Y, Y. So it is again round yellow. What is this one? Round yellow.
Round yellow.
Round.
This one, I'm just talking about the phenotypic character, how they are in appearance. This one,
Round yellow.
round yellow. This one,
Round green.
round green. This one, round yellow.
Round
This one,
Round green.
round green. This one, round yellow. This one, round yellow. This one,
Wrinkled yellow.
wrinkled yellow. Here, wrinkled yellow. Here, round yellow.
Round.
Here,
Round green.
Round green. Here,
Wrinkled yellow.
wrinkle yellow. Here,
Wrinkled
Wrinkled
green.
green.
green. So this will come in the ratio 9:3:3:1. 9 will be round yellow, and these three are new combinations. What are they? Round green, wrinkled yellow, and this is as usual the pure breed but recessive. This is wrinkled green. Okay, so this is how we conclude with Punnett square. This is possible with Punnett square because there the other one is little complicated. Yes. Okay, so what and all did we go through today? We just saw the basics of genetics. What is DNA, what is chromosome, and what is gene. And we saw the types of traits, the traits which are inherited and the traits which are acquired. Then we saw the number of chromosomes in our body, how are they transported, and what is actually done. The gene gives rise to protein, and protein is the one which is responsible for our character. Then we talked about Mendel, why is he called the father of genetics, and what is his contribution. 3 laws were proposed by him, very importantly: law of dominance, law of independent assortment, and law of segregation. And based on what? Based on the cross he did with the pea plants. It was monohybrid cross and dihybrid cross. Apart from that, we also just had a glimpse of sex determination. Am I correct? Yes. Hope the session was useful for you. Any doubts?
Yes.
Any doubts? Clear? Okay. So in the exam point of view, if you see, you will have mainly the cross will be there. The Punnett square will be there. Sex determination is a very important question. And unfortunately, you don't have evolution part this year. So you have to concentrate on every topic of this particular chapter. This much, what we have explained now, only this much is there. Only thing is it has to be a little more precise and improvise properly according to the perfection. That's all. Yes. Okay. Thank you so much for the session.